Hello students, in this class of hormone biochemistry and function, we will study further in the hormones of GI tract and pancreas. We have already discussed about the structure and little bit mechanism of insulin. In this class, we will study about the effect of insulin on metabolism of carbohydrate, protein and fats. So first we'll start with the effect of insulin on carbohydrate metabolism. So carbohydrate metabolism involves all these tissues, muscle, liver, brain, and other cells. So specifically, I'll start with muscle. So insulin promotes muscle glucose uptake and metabolism. So what happens in this case? It has two effects. First, it is showing its effect on exercising muscle. So while it is exercising or muscle is contracting and relaxing, insulin increases the uptake of glucose into the cell. So more and more glucose will start going inside. And apart from that, even a small amount of insulin is sufficient to show its action. Or in fact, muscle are not this time dependent on insulin for their function. So they have to, their permeability to glucose is increased. So they more and more glucose can be taken up. And second condition is after a few hours of meal, after a meal or a few hours. So when your food is absorbed into your gut and glucose concentration is high, so that time glucose start coming into the muscles or by the action of insulin, because insulin increases the transporter on the surface of the cell so that more and more glucose can be internalized. When excess of glucose is there, so what happens? This will be stored in the muscle in the form of glycogen. So this is in the muscle. So this glucose will start forming glycogen via different set of enzymes, and this will stored into the muscle. So 2 to 3% of concentration is the glycogen in the muscle when it is uh, maximum, or when you have excess of glucose, this can store in this concentration. Next is quantitative effect of insulin to facilitate glucose transport through the muscle cell membrane. So this picture shows what is the effect of insulin for the internalization of glucose. So there are two things: on x-axis extracellular glucose, while on y-axis it is intracellular glucose. So if you provide glucose via infusion or by any other mean, so when the concentration of extra glucose concentration is increasing, so it is seen that when insulin is not there though, so there is no effect of glucose concentration on intracellular glucose. While when insulin is there inside your cells or outside the cell, that will increase the uptake of glucose by the cells. This way, so internal concentration of glucose is increased in this case. So you can see there is a plateau in case of control, and there is exponential increase in the concentration of glucose inside the cell. Next is insulin promotes liver uptake, storage and uses of glucose. So this does by liver phosphorylase. Liver phosphorylase is required for glycogen breakdown. So this is not required at this time because you already have a lot of glucose in the circulation. So this is in liver, which is inhibited, and more glycogen will be not forming. And insulin enhances uptake of glucose from the body by the liver cells. So extra glucose will be taken up by the liver cells, and this insulin increases the activities of enzyme that promote glycogen synthesis, including especially glycogen synthase. So this enzyme is required for formation of glycogen. So this enzyme will be activated while phosphorylase will be deactivated. So this way, this will lead to the formation of more and more glycogen. Glycogen is released from the liver between meals. So once your glucose concentration is reduced after few hours of meal, then what happens, all the reactions which we studied till now, they will be reversed. So insulin concentration will be decreased, and this will lead to the glycogen stop in the glycogen synthesis, and glucose uptake will be stopped. Both the things will be stopped, and glycogen phosphorylase will be activated. So glucose is released from the liver between meals. When you have all the glucose utilized for different purposes, it is stored and there is no more uh, high glucose into the circulation. That time what will happen? So this pancreas will stop releasing the insulin. So concentration of insulin will be less in the circulation, and like half-life of the insulin is only 6 minutes. So this will help in the clearance of the insulin from the circulation. While glycogen synthesis, which happens when glucose concentration is high, that will be inhibited, and for that glucose uptake is required. So this will also be stopped because there is less glucose outside the cell. Glycogen phosphorylase, which is required for the removal of glucose from glycogen, will be activated. So this will lead to the increase in concentration of glucose between the meals. So you can see in this picture that glycogen phosphorylase is the enzyme which is removing the glucose 1-phosphate from glycogen, and activation of glucose phosphatase. So in this form, it is getting released into the form of glucose 1-phosphate. So glucose phosphate, what it will do? It will remove this phosphate group from the glucose, and free glucose will be released into the circulation. And this glucose is able to get out of the cell in the circulation. Stored glycogen will be broken down into glucose, and glucose will come out into the circulation. Next is insulin promotes conversion of excess glucose into fatty acids and inhibit gluconeogenesis in the liver. So how it does? So formation of fatty acid from glucose occurs by three different steps. Oxidation of glucose to pyruvate via EMP pathway or glycolytic pathway. This pyruvate will enter into the pyruvate dehydrogenase complex and form the acetyl coenzyme A, and acetyl coenzyme A will form then fatty acid via fatty acid synthase. And then gluconeogenesis will be inhibited by decreasing the enzymes required for the gluconeogenesis, as well as it will decrease the release of amino acid from the different tissues to be utilized for glucose formation. Lack of effect of insulin on glucose uptake and uses by the brain. So brain is highly specific that it can take glucose without interference of insulin. Why? Because it has specific glucose transporters. So brain cells have high permeability to glucose, and it is insulin independent, and it is of utmost requirement to keep the glucose level balanced, because if you have less concentration of glucose, this can lead to the hypoglycemic shock, which will lead to the nervous irritability, fainting, seizures, and coma. So for this purpose, glucose concentration should be balanced. Effect of insulin on carbohydrate metabolism in other cells. So other cells apart from muscle cells, they are also dependent on insulin by the same mechanism. They have to increase the receptor or a transporter for glucose, and so that they can internalize more and more glucose. And on adipose tissue, these are requiring the glucose for the formation of glycerol. So this glucose will be internalized, and here it will form glycerol, and glycerol will then get accommodated into TAG, and this TAG will be stored into the adipose tissue. Next is effect of insulin on fat metabolism.